2.5 billion years ago, the young planet Earth was a mass of cosmic dust and particles. It was almost completely engulfed by the shallow primordial seas. Powerful winds gathered random molecules from the atmosphere. Some were deposited in the seas. Tides and currents swept the molecules together. And somewhere in this ancient ocean, the miracle of life began. The first organized form of primitive life was the tiny protozoan. Millions of protozoa populated the ancient seas. These early organisms were completely self-sufficient in their seawater world. They moved about their aquatic environment, feeding on bacteria and other organisms. They were covered with hundreds of tiny whipping hairs called cilia and flagella that made movement possible. From these one-celled organisms evolved all life on Earth. And the foundation of life, the cell, has endured unchanged since the first tiny organisms swam in the cradle of life, the sea. Every living being is made up of cells, the basic units of life on Earth. And all cells reproduce themselves by dividing into two identical cells. This is a single body cell from a human being. And this is the process by which all cells reproduce. Now, there are two cells, each exactly like the parent. In the cell's nucleus is the extraordinary chemical substance DNA. The DNA is contained in 46 chromosomes in every cell of the human body. Each chromosome, in turn, is made up of thousands of genes, discrete segments of DNA which lie along the chromosome. Penetrating even deeper into the structure of the chromosome, we see the DNA molecules themselves. DNA contains all the genetic information of the cell, and it is the only living substance capable of reproducing itself exactly. Without DNA, duplication, and therefore life itself, is not possible. All DNA in all living organisms is chemically identical, but its arrangement in genes and chromosomes determines what the cells will become. Here, a tiny primitive organism, the protozoan. A massive complex mammal, the elephant. A palm tree swaying in the tropical breeze. Or a human being. We are about to witness, for the first time, a wondrous and complex process. The actual conception and growth of a new human being. Each beginning lies deep inside the mother's body in the ovaries. Each ovary contains 250,000 egg cells, which reach full development even before the woman herself was born. The eggs each contain 23 chromosomes, the mother's genetic contribution to her future offspring. During each menstrual cycle, the ovaries produce hormones which stimulate the growth of a single one of these eggs. At the midpoint of her cycle, the follicle, which encloses the egg in its protective layers, ruptures. This is ovulation. The egg travels through the fallopian tube, which connects the ovary and the uterus, or womb. In the tube, the egg waits for sperm from the father to fertilize it. The fertilized egg, now with genetic material from both parents, moves through the tube to the uterus, where it attaches itself to the nutrient-rich lining. And here, the ovary itself. In cross-section, stained blue for better visibility, we see the hundreds of tiny egg cells. This one is nearly mature and is surrounded by its nutritive layers. When it is fully developed, the follicle which encloses the egg will swell, like this one, with fluid, the liquor folliculi. Inside the follicle, the egg and its nutritive layers floats in this water of life, which has the same salt content as the sea. The swollen follicle here on the left is then gently brushed by the swaying fimbriae, the outermost fringes of the fallopian tube. These fringes are activated by the hormones just before ovulation. They sweep over the surface of the ovary, searching for the newly released egg. As ovulation nears, the fringes become filled with blood. The follicle at the center is about to burst. Directly above the white reflections, the liquor folliculi pours out of the follicle. Concealed somewhere within it is the egg. The fringes search for the newly released egg. It lies here, surrounded by a cloud of its own nutritive cells. It is only the size of a grain of sand. At the far right, the egg is drawn into the fallopian tube. Deep inside the egg is the nucleus of new life itself. This is the mouth of the fallopian tube. Its almost imperceptible muscle contractions move the egg along toward the uterus. Here, the egg has reached the interior of the tube itself. These many folds of tissue are lined with tiny cilia, which maintain a constant, gentle motion that draws the egg along its length. The inside of the tube is actually only about twice the thickness of a single human hair. It takes the egg 3 to 4 days to travel this distance of only 5 inches. The translucent egg here lies in a muscle fold deep inside the fallopian tube. On the diagonal edge below the egg, we can see a thin, shimmering layer of the cilia which line the tube. Gently moving the egg along, they are exactly like the cilia of the primitive protozoa. The egg must join with a sperm within 24 hours of leaving the ovary for conception to take place. If no sperm are present, the egg disintegrates, and the same cycle will happen again the following month and throughout all the woman's childbearing years. This remarkable cycle is made possible by complex molecules, the sex hormones. Crystallized and stained, they look like this. Hormones control a woman's entire reproductive cycle. Hormones control a man's reproductive ability as well. Only 7 weeks after conception, the fetal system is permanently sensitized to be male or female by the sex hormones. One fetus will develop ovaries, and its brain will be programmed to release hormones in cycles. It is female. Another fetus will develop testicles, and its brain will release constant low-level amounts of sex hormones. It is male. "77 billion human beings have lived on this planet. 4 billion live here now. And every year, 128 million new ones are born. We have already seen part of the woman's remarkable contribution to this unending process. Now, we will explore the man's. During a man's lifetime, his testicles produce billions of sperm. They are stored here in the epididymis. The Cowper's gland produces a lubricating fluid to aid the sperm's journey. The prostate gland secretes an alkaline fluid to protect the sperm. Together, they produce most of the fluid called semen. During ejaculation, the sperm leave the epididymis, moving through the vas deferens, this tube in the body cavity. They approach the seminal vesicles, which release a solution of sugar to nourish them. Cowper's and prostate fluids combine with the sperm, and the blended semen continues out of the man's body through the urethra in the penis. This is the interior of the urethra itself. We are traveling up into the man's body from the outside through the penis. The red areas are blood vessels. The black spots are calcium deposits. In reality, the journey is only 7 or 8 inches long. Ahead, the red area is the opening to the bladder. During sexual arousal, a valve here stops urine from entering the urethra. The valve is now open. Here, near the bladder, the urethra passes through the prostate gland. The walls of the prostate have more than 30 orifices, like these, which squeeze out prostate fluid when sperm pass through the urethra. It is about the size of a golf ball, and it resembles a sponge. Inside, it is filled with the small cavities which produce the fluid. These are the testicles, highly magnified and without their usual protective pouch. Unlike the ovaries, their counterpart in women, the testicles lie outside the body cavity. They are made up of the tightly coiled seminiferous tubules, which have a total length of 700 feet. In the tubules, sperm are produced at the extraordinary rate of 100 million every 24 hours. In the tissue between the tubules, the sex hormone testosterone is produced. The testicles are the essential male organs of reproduction. They function well only under very specific conditions. One of these specific conditions is temperature. Sperm are produced most efficiently at several degrees below normal body temperature. These images from a thermal camera reveal the various temperature zones in the scrotum, the pouch of skin in which the testicles are suspended. The white areas are the warmest, and the green ones the coolest. The scrotum keeps the testicles away from the body's heat. This is a cross-section of one of the tubules in the testicles, stained blue and magnified 2,000 times. At the dense center of the tubule is the transport canal, which carries mature sperm away to the epididymis for storage. The smaller compartments produce many new immature sperm every 36 hours. As the sperm become more mature, they move closer to the central canal. The average man produces over 400 billion sperm in his reproductive lifetime. These sperm, magnified 4,000 times, have been in production for approximately a month. They are tightly packed in the tubule within the testicle, their heads and tails intertwined. Inside the developing sperm heads, the normal body cell's 46 chromosomes have been reduced to 23. But this reduction of chromosomes, so essential to future reproduction of the species, puts the sperm in mortal danger. The man's body considers these cells enemies. But the sperm are defended by nurse cells, like this one, and the large cell at the right. They form special protective barriers around the maturing sperm cells. Each nurse cell cares for many sperm at a time. This tendril is part of the remarkable communication network which connects each nurse cell to over 150 separate sperm cells throughout the tubule. The tendrils enable the nurse cells to feed and protect the maturing sperm and to move them closer to the transport canal. This sperm is nearly mature. During its development, it has lain in total passivity in the tubule, being protected and nourished by the nurse cell. When mature, like this one, it is transported by the nurse cell to the epididymis. Millions of sperm are stored in these densely packed tubules, which, if stretched out, would be 15 or 20 feet long. The mature sperm in the epididymis will pass out of the man's body through ejaculation 200 to 300 million at a time. Or they will eventually die and be reabsorbed. These sperm carry the man's genetic material, and they are fully mature. But they are not yet able to fertilize a woman's egg. They do not attain that ability until they are actually far up inside the woman's body. They may stay in the epididymis, the bag-like structure on the left, for several weeks. While here, they develop the ability to swim. A sperm propels itself by means of a long tail, a flagellum, which lashes vigorously from side to side. This extraordinary activity has but a single purpose: to deliver the genetic material in the sperm's head quickly and precisely to the woman's egg. On the tip of the sperm's head is a layer of enzymes and enzyme inhibitors. The inhibitors preserve the enzymes, which will be needed later to penetrate the surface of the egg. This is how the sperm's head looks without its enzyme cap. The material of the head itself is loaded with the actual genetic information which will be transmitted to the egg. These are the strands of DNA which will be transformed into chromosomes. This is one of the many fuel packets arranged along the midpiece of the sperm. The fuel consists of sugars, which is converted into energy for the sperm's locomotion. The propulsion system of the sperm's tail is made up of a bundle of cords or filaments. The filaments are covered with thousands of tiny hooks. As the hooks intermesh, the whole bundle begins to bend, and the tails of the sperm begin to whip around like this. It is a primitive but highly efficient system of propulsion. The system does not function perfectly every time. On the average, up to 20% of a normal man's sperm are deformed or imperfect in some way. Perhaps one reason why a man produces so very many sperm is that so very many things can go wrong with the sperm themselves. They may have 2 tails, like this one. Some even have 3 tails. In another common deformity, the heads of these sperm appear to be nearly severed from their tails. This sperm has been attacked by bacteria. And this one may have been produced too rapidly. It will never complete its development. White blood corpuscles, the large round cells, are often found together with defective sperm. Their presence indicates the likelihood of infection, fever, or even a common cold. Most of these sperm deformities could have been caused by just a slight elevation in the temperature of the testicles. Environmental conditions, too, often damage sperm production. Overcrowding, stress, smoking, chemical pollution of air and water, occupational hazards, radiation, poor nutrition, all part of modern life. Even our modern clothing may affect sperm production. Tight blue jeans, for example, hold the testicles up close to the body, perhaps raising their temperature those few critical degrees. But in spite of such obstacles, human beings have been successfully reproducing for millions of years. Since the first organisms swam in the sea, the cycle of life has been continuous, fueled by the need and the drive to reproduce. Among human beings, that drive is intimately linked with attraction and desire. Affection and romantic love may initiate the dance of courtship, which may lead to conception. Sexuality, affection, and tenderness can all be part of the complex rituals of mating. But desire and physical love possess a silent language all their own that signal partners' readiness to each other. The eyes are part of that silent language. Visual impressions stimulate and excite special nerve cells in the sexual areas throughout the body. The skin, too, is part of the silent language. It is amazingly sensitive and responsive to the touch. It contains millions of these sensory receptors, which, when stimulated, direct messages of sexual arousal to the brain. Many of these are concentrated in the body's most responsive areas, the erogenous zones. One of these, the man's penis, is densely packed with sensory receptors, here seen at enormous magnification. They relay messages of sexual stimulation to the brain, which triggers the complex sequence of events leading up to ejaculation.